So first, I'd really like to thank Nancy and Jeff and George Church and Autodesk and the other organizers. I think this is such an exciting time for the field, and this is such a great group of people to collaborate with, and I'm really excited to see what happens over the next decade. So I thought today I'll just tell you a little bit about the research going on in my laboratory with a little emphasis on cell-cell communication. So I originally trained in organic chemistry, so I did round bottom flask chemistry, and then I did chemical biology, which sort of combines that with molecular biology. But when I started my independent career, I came back to Columbia in 1999. I thought that, there were already a lot of people doing beautiful work in that kind of chemistry, and I thought instead it could be fun to see if we could engineer yeast to do new chemistry, and I think that has turned out to be a very good decision for us. And when I started, specifically what we started with is new technologies, and so we basically asked, could we engineer yeast to carry out both the mutagenesis and the selection steps of directed evolution directly in the cell, so we could in essence program the yeast to engineer the new chemistry for us. And I think if we think about how hard it is and how large of a library it still takes to engineer an antibody, I think it really makes sense that we are going to need new enabling technologies. So we started by focusing on the selection problem in the early 2000s. We showed we can combine basically yeast genetics and organic chemistry to trick yeast into to carrying out new chemistry. And I think, when we published this paper in 2002, I think our H index was really low. But I think maybe this makes a lot more sense here at the context of this meeting. We then went on to work on the mutagenesis step of directed evolution. And this was work carried out by Dante Romanini and Pamela Peralta-Yaha. And we basically came up with a way that we can have recombination-based mutagenesis that's compatible with sexual reproduction. And we have a heritable cassette plasmid that keeps track of the mutation we've made and also allows us to pass on to daughter cells. And while we've only done a little work with this technology at this point, I think if we think about, again, the kinds of library sizes that might be required to engineer multi-component systems, if we can take advantage of sexual reproduction like nature does, arguably, rather than just searching a library of 10 to the 8th, if we had a virtual search, by analogy to computational chemistry, of 3 libraries of 10 to the 8th each, and they could meet the winners of themselves, this could, maybe be a virtual search of a library of 10 to the 24th, which gets exciting for engineering a system. But I think really what we and others are really excited about now is sort of, as we've had a series of enabling technologies in the field of synthetic biology, is to really ask the question, if we can engineer a cell to do anything, what would we like to engineer it to do? And I just like to emphasize that we don't want to engineer yeast to do something like, synthesize a molecule that it then leaches out of the cell. We're really interested in applications where the yeast itself, as opposed to a small molecule or a biologic like an antibody, is the product. And so that's, really the question that's on our mind. And we've chosen to work with yeast, which makes a lot of sense, both because it's fantastic in the laboratory and it's already used as a household product, and it's safe for centuries. So I think really the first application we thought of with yeast is in the area of diagnostics. and basically I think the idea is very simple. If we think about antibody dipstick tests, those are actually pretty expensive. They're about $1 a test, and they can require, depending on the format, expensive reagents or equipment to read them out. And so we basically ask, could we engineer dried yeast to do the same thing by putting a modular receptor on the outside of yeast and having the yeast make something colorimetric, so that even somebody in a remote village can just detect it by eye. And we estimate that dried yeast could be about 1 cent a test, and again, really not requiring any technical reagents or expertise. And I won't go into this project today. I'll just mention it's under review, but this was really a collaboration between Neely Ostrov, who's here at the meeting,
Sonia Billerbeck, who's here at the meeting, and also Miguel Jimenez. And suffice it to say that we have been able to engineer our first generation biosensors. We started off by focusing on fungal pathogens, both in human health and in food safety. So here we're sort of thinking about engineering an individual yeast, as a product. But I think one of the next things that, we're going to want to do is engineer communities of yeast and other organisms that can function together to carry out more complex functions. And this project has been a collaboration with Jeff Boca, and it's been carried out by Sonia Billerbeck, Miguel Jimenez, Jamie Brisboe, Netta, who just spoke, and Michael Shen. So if we think about having a community, it naturally comes to mind that we'd like to have some kind of communication language. And I think it's fair to say that really the best communication language that is out there comes from bacterial communities where they use like acylserine lactone molecules. But the problem is those just aren't very scalable. And even though they've been around for a while, we've only really seen 2 to 3 orthogonal systems. And so we basically said, look, yeast actually already has a orthogonal communication language, and this is using peptides as the signaling molecules and GPCRs as the sensor. And so we hypothesized that we could just go into the, what's known from the fungal databases from sequences, and we could just pull out the sequences of the peptide and the GPCRs for the different fungus for their communication language. And so suffice it to say, we worked out a pipeline to do this. We now have cloned over 30 peptide GPCR pairs. We find that they cluster. About 2/3 of them are highly orthogonal to one another, and the other third, interestingly, have a lot of crosstalk. So that gives us, immediate access to much bigger libraries than we've had before, in essence. For the peptide module, we also have a sort of a framework for how we carry this out. And then we developed assays for being able to see if the peptides are being secreted. So suffice it to say, again, we have on the order of 30 peptide GPCR building blocks at this point. And really what we're doing now is starting to play with communication. We've been able to demonstrate successful communication between 2 yeast, and we're beginning both to scale this and to look at like a daisy chain motif or other ways in which we might have these yeast communicating. But I think this is just the beginning. I think this really can be a very massively scalable communication language that can be easily imported, hopefully even into mammalian systems with the use of a GPCR. And I just want to end. I hope I highlighted some of the individual work, but I'm very lucky to carry out this research on the 12th floor of a new interdisciplinary science building at Columbia, and I'm in both the chemistry and the systems biology departments at Columbia University. Thanks so much. Is everybody ready for a snack? Okay, I think I get to decide then. So it's the close of this session, and we'll see everybody in the break area. Great.